The next domain in the tree of life is the Archaea. While they look very similar to bacteria, they are actually genetically very different. That's why they're grouped into their own domain. Bacteria and Archaea used to be grouped together until we were able to compare their DNA in the 1970s. We discovered then that Archaea are actually as different from bacteria as humans are. We spent a lot of time talking about bacteria, but we will spend less time talking about Archaea. That is because we know much less about Archaea than we do bacteria. At first glance, Archaea and bacteria are very similar. It's no surprise that until fairly recently, we didn't even know they were separate groups of organisms. Both are prokaryotes, so they lack a nucleus and other membrane-bound organelles. Both are unicellular organisms that can live individually or in colonies. In both bacteria and Archaea, some species have a tail called a flagellum that they can use to propel themselves, though not all of them will have this. The cells of both bacteria and Archaea are enclosed in a plasma membrane, and many, but not all species in both groups often have a cell wall. And like bacteria, Archaea are also very diverse and found in a wide range of habitats. They are common in soils, and there are most likely Archaea living in your digestive system right now. But when we developed the tools to look at the genetic and molecular makeup of these organisms, we discovered that bacteria and Archaea are actually very different, even though they look similar. Again, it wasn't until the 1970s that we started doing genetic work and discovered that Archaea are genetically distinct from bacteria and actually are an entirely separate domain. So the two groups are as different from each other as either are from the eukaryotes. In addition to these genetic differences, we discovered that their molecular makeup is very different. While their plasma membranes function similarly, they are made up of different types of lipids. And there are also a lot of other molecular differences, such as the types of enzymes they produce and how their cells function. While many bacteria are pathogenic, meaning they can cause disease in other organisms, no Archaea have been found to cause disease. And Archaea are also distinct from bacteria in their ability to inhabit some very extreme environments. This is one of the reasons we don't actually know that much about Archaea yet. Remember, we didn't even have a name for them until the 1970s, so it hasn't been that long that we've been able to study them as a distinct group. But also, many Archaea live in extreme environments that are difficult to replicate in a laboratory. So because we can't just grow them on a culture plate, they're very difficult to study. So what are some of these extreme environments that Archaea inhabit? One example are Archaea that live in hydrothermal vents in the deep sea. These hydrothermal vents release very hot water, and temperatures can be as high as 400 degrees Celsius or 752 degrees Fahrenheit. So these species of Archaea are adapted to handle temperatures that would kill most other living things. At temperatures above 40 or 50 degrees Celsius, the proteins in most other organisms begin to denature, killing them. These Archaea are also adapted to the extremely high pressure of these deep sea environments. Other Archaea can survive extremely strong acids or extremely high salt concentrations, which would kill most bacteria. Some Archaea can live in water with a pH of 0, which is the same pH as hydrochloric acid, and others can live in extremely salty water. If you think back to when we talked about concentration gradients and diffusion, for most other living cells, these very salty environments would cause water to rapidly leave the cell, causing it to shrivel. So it's pretty impressive that these Archaea have developed adaptations to survive these types of conditions. We call these species adapted to extreme conditions extremophiles. And again, living in these extreme types of conditions makes Archaea very difficult to study in the lab. You can imagine that it may be very difficult to replicate the pressure or temperature of a deep sea vent in the laboratory. Because of this, we've only been able to culture and grow 1/4 of all archaea that have been identified. So we still know very little about most of the archaea that are out there. And there are probably a lot of species we haven't even identified yet. So if you want to discover new species and learn about a still very unknown group, studying archaea would be a great way to do that. Now pause the video for these last 2 participation questions.